Hello.
Morning.
Morning. How are things?
Good.
Yeah.
Or, I was gonna, well, you tell me when you're getting started.
Oh, yeah. I mean, we can start whenever, I guess.
Um,
I'll just do a little intro here, and then we can go ahead.
Sure.
All right. So, yeah, welcome to the meeting. Again, we this week we had one meeting, and I actually had an admin meeting with Hussein, but we had the Diva Worm meeting. So the Diva Worm meeting is actually quite good this week. We had two guest speakers, actually two speakers going over their things. I had Kirill, who is someone who works at NVIDIA, and he's been working on some biological simulations of development. So he's been working on some things that involve biophysics and cell division and visualizing the embryo. And then Shreyas, who is sort of, maybe a GSOC hopeful this year. Um, and he's been working on the temporal graphs. So he's been working specifically with neural ODEs and applying that to connectome data in C. elegans. So that was really an interesting set of slides from him. So, he's been giving updates. This is his latest update. Kirill gave a talk on his simulation work, and then we had a discussion about that. And then before those two talks, I talked a little bit about an article from a popular website where they were talking about sentience and how to model sentience in C. elegans and what sentience means. And it goes a little bit into the philosophy literature and a lot into speculation, but it was really interesting. Is the Asimov Press article that we talked about in the meeting. So that's all on the DevoWorm YouTube channel. And then, of course, we are thinking about this coming year. So we need to think about what kinds of things we want to do, what kinds of papers we want to write, what kinds of events we want to attend, et cetera, et cetera. So if you have ideas, please let me know. And maybe I can meet one-on-one with you to discuss things further. All right. So I think that's a good recap of the week. And so I know Morgan has probably been pretty busy this week.
Well, yeah, I'm blanking on the Asimov Press guy's name, but it was really interesting hearing him speak and giving, both his kind of how he came to be running a press.
Oh yeah.
And and his background in synthetic biology. So someone who's definitely like at a top biolab for both understanding molecular biology as well as synthetic biology, but then talking to someone about being able to have the freedom to write the kinds of things that he was interested in writing.
Yeah.
And starting a press, and so really, really interesting. And I think that's why you see like the articles that they produce are just so interesting, because it's like, wait, once you have the backstory, it's like, oh yeah, okay, this is a place that allows this kind of creativity, or kind of allows people to pursue their passions. Anyway, I was related to that. I wanted to see if you had covered the state of brain emulation report.
Is this a new thing that just came out, or?
Yeah. So,
Okay.
this is I mean, the last two authors I recognize. I mean, I've met Philip Shue. So that's the guy. He came out of Kristen. I'm blanking on her last name. He came out of the Berkeley lab that was doing really interesting Drosophila, connectomics.
Oh yeah.
And then he was doing kind of neural modeling using the connectomics.
Right.
But he was doing it where it could run on a laptop.
Oh yeah.
But it was using the whole connectome, but it was also very, I think it was like the particular the particular model, because he wasn't using a the kind of model that like Neuron, the C++ package, would use.
Right.
I'm blanking now on that particular model name. It's like
I think we covered it in the meeting once.
Yeah,
but I can't remember the name either.
I can find it, but
Yeah.
but the other senior author is Anton Arkhipov, the Allen Institute. So this is the developer of I forget what he calls his package. So this is an alternative to Neuron, the C++ package. The poorly named for SEO purposes C++ package. What's his package called? But because they just had the meeting. They had like the AAI meeting in Singapore.
Mm-hmm.
That was like AAI NeuroAI meeting in Singapore. I'll find it.
Okay.
But yeah, so this is Philip Shue now works for the for this company called Eon Systems that had come by that they'd actually been looking for space in the tower. But it starts with C. elegans.
Okay.
And but then covers larval zebrafish, Drosophila, mouse, and human. Right. So our analysis focuses on 5 selected model organisms, offers only a glimpse into the vast topics of neuromodulation and neuroplasticity, and deliberately omits detailed discussions of transcriptomics or proteomics. But it's very much, trying to cover as much of what I would say was like very much his PhD or postdoc work, which is like, if you're able to have a connectome, can you kind of breathe life into it with a neural model, with a computational neural model, right? And yeah, sorry, I'll find the link to it. There you go. I mean, that's the archive. I'm trying to find where they actually sent out the. Oh yeah, this Maximilian Schon's. Yeah, here's the fancy one. Is this the second link? I just thought, especially because it's covering the like they got Shu et al. 2024 talking about the release of firework. Oh yeah, okay. The model that he employed in that paper was a leaky integrated fire framework, and that was why he was able to run it on a laptop, even though it was using, a very large connectome. Instead of like trying to do a Hodgkin-Huxley model, because I think like a couple of years before that they'd done a big supercomputer Hodgkin-Huxley model, and yeah, like the computational demands were just disproportionate. Anyway, I thought it would be interesting.
Yeah.
To they bring up OpenWorm. Longtime OpenWorm lead Stephen Larson. And I know this definitely has Foresight Institute funding, and of course, Ed Boyden and Conrad Cording are pushing, a renewed effort in collecting data from C. elegans to do this kind of brain emulation work.
Yeah, that sounds interesting. Why don't we go over it? I'll go over
Sure.
the links. So yeah, just let me know if you want to talk about these things. I'll pull them up one at a time here. So the report is here. So this is Adam Marblestone and Jiangfeng Feng. Yeah.
Yeah. So Anders is someone you will see at most of the Foresight meetings. Yeah, and he has he also has a great like super in-depth paper that he did with Logan Collins.
Oh yeah.
And they've got a proposal for like a next gen X-ray microscopy.
Yeah.
For connectomics. He's got a package called Brain Genix, like GENIX, I think. Anyway, that like again, this is a group of people that have definitely been working on this for a long time. Like, both kind of collecting, how to get the connectomics, right, at like, nanoscale,
Mm-hmm.
and then how to turn that into a computational model.
Yeah. So it looks like we have the state of brain emulation report. Yeah. Why don't we look at the summary?
How about this, the summary and then the budget guesstimator?
Oh yeah. All right. So these are the executive materials. Just kind of going over what they mean by brain emulation. So accurate brain emulators would occupy a unique position in science, combining the experimental control computational models with the biological fidelity needed to study how neural activity gives rise to cognition, disease, and perhaps consciousness. A brain emulation is a computational model that aims to match a brain's biological components and internal causal dynamics at a chosen level of biophysical detail. Building a brain emulation requires three core capabilities. So we need recording of brain activity, we need reconstruction of brain wiring, and we need to digitally model brains with respective data. In this report, we explain how all three capabilities have advanced substantially over the past 2 decades to the point where neuroscientists are collecting enough data to emulate the brains of sub-million neuron organisms, such as zebrafish larvae and fruit flies. So these are connectomes with under 1 million neurons, basically. The first core technique required to build brain emulations is neural dynamics, in which electrodes are used to record how neurons, from a few dozen to several thousand, fire. Functional optical imaging transitioned from nascent technology to large-scale recordings. So examples are calcium imaging, voltage imaging, and related technologies. So calcium imaging is where genetically encoded indicators report correlates of neural activity. And then voltage imaging resolves individual spikes in tens of thousands of neurons. And in the calcium imaging, they have examples from mice, and larval imaging or voltage imaging, they have examples from larval zebrafish. So taking neuron count and sampling rate into account, these improvements represent about a 2 order of magnitude increase in effective data bandwidth of neural recordings in the past 2 decades. Then they have causal perturbation methods. So these are things like optogenetics, where you can have a signal that modifies brain activity, and if you do this in a patterned way, you can understand the effects of that. So this is where we want to be able to reverse engineer neuron level input-output relationships across the entire small nervous systems. So despite this, neural activity recording still faces significant trade-offs across spatial coverage, temporal resolution, recording duration, invasiveness, signal quality, and behavioral repertoire. Even more challenging is recording of modulatory molecules like hormones and neuropeptides. Defining whole brain is capturing more than 95% of neurons across 95% of brain volume simultaneously. No experiment to date has delivered that scale with single neuron, single spike resolution in any organism during any behavior. So this is kind of what they need, is this idea of the whole brain. They need more than 95% of neurons across 95% of the brain volume, and I assume for a significant period of time. So we don't have that right now, but that's what we're trying to aim for. And so that is sort of the neurophysiology. And then the connectomics, which is the second core technique, is used to reconstruct wiring diagrams for all neurons in a brain. Connectomics models have moved past C. elegans worm brain mappings to produce, more recently, 2 fully reconstructed adult fruit fly brain connectomes. These are the motor and the sensory connectomes, the ones that are published currently. We've gone over these in previous meetings. These are, of course, both larger than the C. elegans connectome, and then, of course, you can put them together to understand, basic sensory motor behaviors in Drosophila, although it's not all of the neurons. It's just kind of two large subsets of neurons. So this is a big achievement because fruit flies have about 3 orders of magnitude more neurons than a C. elegans worm. Several additional scans in other organisms, such as larval zebrafish, have also been acquired and are expected to complete processing in the near future. Dataset sizes now increasingly reach petabyte scale, which challenges storage/backup infrastructure, not only with costs, but also the ability to share and collaborate. So this is, despite sort of the scaling up of the method, it is indeed faster to make connectome maps today than it was just a few years ago, in part because of how the actual images are acquired and stitched together. Progress is being enabled by a mix of faster electron microscopy, automated tissue handling pipelines, and algorithm image processing / neuron tracing. Each of these improvements have contributed to push costs per reconstructed neuron from an estimated $16,500 in the original C. elegans connectome to roughly $100 in recent larval zebrafish projects. Proofreading the manual process of fixing errors from computerized neuron tracing remains the most time and cost-consuming factor. And this is going to be even more true for mammalian neurons, where you have larger sizes, complex morphologies, and more neurons. Experts are optimistic that machine learning will eventually overcome this bottleneck and reduce costs further, but as of now, reconstruction efforts are basically limited to contour tracing, to reconstruct wiring diagrams, and lack molecular annotations of key proteins and other molecular aspects of the cells, limiting their direct utility for functional interpretation and computational modeling. Many experts are optimistic that in the future one might be able to build connectomes much more cheaply by using expansion microscopy rather than electron microscopy. This is a technique that Ed Boyden, I think, has done, where they blow up a piece of tissue with some sort of method, and then they can expand the tissue. Obviously, it's you have to have dead tissue or like a sacrificed animal to do that, but you get the tissue expands, and so you can increase the resolution of your imaging technique by a fair amount. And so this will help us understand maybe some of these molecular annotations. And then, of course, we have other methods such as protein barcoding for self-proofreading, so we can use those as well. The final capability of this is computational neuroscience, or the ability to model brains faithfully. The capacity to simulate neural systems is advanced, enabled by richer datasets and more powerful software and hardware. In C. elegans, connectome-constrained and embodied models now reproduce specific behaviors, while in the fruit fly, whole brain models recapitulate known circuit dynamics. At the other end of the spectrum, feasibility studies on large GPU clusters have demonstrated simulations approaching human brain scale, albeit with simplified biophysical assumptions. They don't have a citation for that. I don't know if they have like a specific study they're talking about or what. On the hardware side, the field has shifted from CPU supercomputers to GPU accelerators. For mammalian scale simulations, the primary hardware bottlenecks are now hardware memory capacity and interconnect bandwidth. On the software side, improvements come from automatically differentiable data-driven model parameter fitting, efficient simulation methods, and the development of more rigorous evaluation methods. Still, many biological mechanisms like neuromodulation are still largely omitted. A more fundamental limitation is that models remain severely data constrained. And so we don't have a lot of experimental data compared to what we need, and we need more individuals, and that's where we have this lack of detail. And of course, we have passive recording, so we don't have naturalistic context that we would like. And then in the conclusion here, they basically lay out the challenges. So in the next phase of research, we need to determine which biological features to focus on. So there are all sorts of things that define connectomes. So you can have gap junction connectomes. You can have chemical connectomes. You also have other types of things like neuromodulators and glial cell relationships that are also important in the connectome. And so these are probably needed to produce faithful brain emulation models. But we don't really have the data or the computational sort of power or representations to do that. Empirically answering such questions calls for more comprehensive evaluation criterion to include neural activity prediction, embodied behaviors, and responses to controlled perturbations. Second, there is a widening gap between our ability to reconstruct ever larger connectomes and are much more limited capacity to record neural activity across them. This discrepancy necessitates that the neuroscience community develops better methods to infer functional properties of neurons and synapses, primarily from structure and molecular data. So these sub-million neuron organisms present a compelling target because you can sort of face both of these challenges on a smaller scale. And so comprehensive functional structure or molecular data sets are attainable at scale, making it possible to empirically determine which biological details are necessary for a faithful emulation. Furthermore, the cost-efficient collection of aligned structural and neural activity datasets for multiple individuals provides the essential ground truth for developing and rigorously evaluating methods to predict functional properties from structure alone. The evidence this generates, defining what is needed for emulation, and validating methods that infer function from structure will be critical to guide and justify the large-scale investments required for mammalian brain projects. So they want to do like a test run in these sub-million neuron systems and then move up to mammalian projects. So faithful emulation of small brains is a necessary first step towards emulating larger ones. And that is kind of the overview. And then they talk about sort of the organism scale. So they talk about C. elegans, larval zebrafish, Drosophila, mouse, and human. They're focused on the adult in all of these except for in the larval zebrafish, of course, which is 5 to 7 days post-fertilization. Has sort of a nervous system that's forming. You can define the nervous system there, but it's not the adult nervous system of the zebrafish. So it gives us a decent-sized connectome without being over 1 million neurons. And so they have the synapse count. So obviously, C. elegans is, pretty tractable. We know that it has about 302 neurons in the connectome, and then 5,600 chemical synapses and 600 gap junctions. So that's even there. That's pretty tractable. Drosophila, if you move up to Drosophila, that has 5 to 10 to the 7th neurons or synapses, and 1.4 to the 5th neurons. So that's quite a big jump in terms of the neurons being the sort of the nodes in a network, the connections being the synapses, and you can see how that explodes when you get to Drosophila. For mouse, we have 10 to the 7th or 7 to the 7th neurons, and for humans, we have 8 to the 6th to the 10th neurons. In neither case do we have fully quantified synapse counts, but we have to work sort of on cubic millimeter blocks and kind of estimate for that. So in the mammalian connectomes, a lot of this is estimated. And then, of course, recording brain activity, you have single cell resolution, you have things like, whole brain imaging or tissue imaging or things like that. And it's a matter of mixing those two things together. This quote here, despite impressive progress in neuron recording capabilities, neuroscience has not yet achieved whole brain recording at single neuron resolution in any organism. And then this is a heat map showing studies that I guess show, what is this? This figure plots the relative distance from the respective organism's maximal value in a set of recording dimensions for a given publication. So I guess this is like if you have a greater amount of neurons, and this is like these different studies here we have for C. elegans, Drosophila, mouse, human, and zebrafish larvae. And for these different dimensions, temporal resolution, duration, resolution, and neurons. So for temporal resolution, we have a couple of studies in C. elegans, and then a lot of studies in zebrafish larvae and Drosophila that have higher temporal resolution. For duration, Drosophila, mouse have longer durations. And then there's one study in C. elegans, and then no studies in zebrafish larvae or humans that are of significant duration. For resolution, we have a lot of good data for humans, a couple studies for C. elegans, Drosophila, and mouse, and then nothing for zebrafish larvae. And then neurons themselves, we have a lot of high resolution data for humans, and then maybe less for zebrafish larvae, mouse, and Drosophila, and even less for C. elegans. So this is neural dynamics recording data coverage. So I'm not really sure of the full details of this heat map, but it's interesting to compare the taxa and to compare the different studies that have come out on this. And so then this is neural recording information over time, information rate over time. So this is year. So this is like the year of the study, I guess. And then on the y-axis is information rate, which is the number of neurons times the frequency. So this is capped at 200 hertz. And so this shows two methodologies, electrophysiology, which is patch clamp or something else like that, and then fluorescence imaging, which is like voltage dyes and things like that. So this is, the fluorescence imaging kind of came online in the 2010s. So you have some studies here. Well, most of the electrophysiology goes back to 1960. And it looks like a lot of this is, I guess, in C. elegans. I don't really know how this is organized, but this line here is C. elegans. And then this line is fly. And I guess we have a lot of studies in C. elegans, and then some studies in fly, and then nothing for mouse, from what I can see from this graph. I don't really know how this is organized with respect to these labels, but basically, we're getting data, we're getting greater coverage, we're getting more information per neuron, and we have more methods coming. So that's the idea. Then they kind of discuss connectomics and how much it costs to acquire connectomics data sets. And so this is, again, connectomics is where you're characterizing all the neurons in a nervous system, you're finding the connections, and you're mapping those out. And that requires a lot of work in terms of segmenting cells, in terms of finding the synapses or the gap junctions in microscopy images, essentially. Sometimes, those maps are going to be pretty easy to build with modern technology. Other times they're very hard to build because you have a lot of neurons. You have, it's hard to find all of the synapses and gap junctions. And then, of course, you have your physiological data, because you can get a static connectome, which is just from microscopy images, like bright field images or fluorescent images. But then you have to, then you have the neurophysiology on top of that, where you have functional data. And so that's, bringing that all together and storing those data are quite challenging in terms of, computational storage and power and all that. And of course, if we're moving up to mammals, then we're dealing with a lot more data. So a mouse brain at 10 nanometer isotropic resolution would require, without further lossless or lossy compression, approximately 1 exabyte of storage. Well, a human brain would require 1 to 1.4 zettabytes. And so you obviously need a lot of storage, a lot of computational power to deal with these types of data. And it's a lot. So this is a huge challenge. So this makes mammalian scale connectomics technically challenging, but not fundamentally limited in the way that whole brain functional imaging appears to be. So this is like, with whole brain functional imaging, we have resolution issues. We have other types of issues with time scale and things like that. But with connectomics, we may not have those hard limitations. So this is a figure table 2. This is synaptic resolution for electron microscopy and reconstructing connectomes. So this is just kind of showing some of the studies over time. So we have the original composite C. elegans connectome. This was done using electron microscopy. This is at synaptic resolution. This is the study from John White in 1986. This is sort of the original connectome study.
When they use the term synaptic resolution,
I guess where you can detail the synapses, because electron microscopy.
I mean, but what they mean by that is actually, I mean, I think it's somewhat misleading.
Okay.
Right.
Yeah.
Or like, I think they're trying to say, you know, neuron 1 is connected to neuron 2.
Right. Right.
Right. But by using synaptic, right, like it you know, the synaptic cleft is quite
diverse. Yeah.
Narrow.
Yeah.
Right. And like, you see what I mean? It's just
it's...
by using synaptic as opposed to something like a connectivity graph.
Right.
You know, I mean, like they don't have to use the. Anyway,
they're implying biological function when they should be implying.
Well, they're implying like you're seeing the synapses. You know.
Well, yeah.
Like I mean, they do. Sorry, I think you're resolving the synapses.
Right.
In a way which isn't fully accurate. Like you're resolving the connectivity.
Right. Yeah.
You know, that we know is or you know, like,
we know it's there, but we don't know how it works necessarily. Or
well. I mean, I just want to also get at like the people who are resolving the neurotransmitter receptors.
Okay.
Right. So you know, it comes back to this point where you might think the neurotransmitter receptors are static.
Right.
And whereas they're not.
Right.
And it doesn't even mean they're neither static in location nor static in number.
Right. They move. They change over time.
They change.
Yeah.
You know,
remodel.
Like the cell is totally in control of changing.
Right.
You know, of changing the proteins that are in its membrane.
Right.
But that you know, and it just comes back to this point. Sorry, I know it's a bit of an aside, but it's just like I think sometimes that they absolutely recognize this,
right,
but they really don't dwell on it.
Yeah, you don't want to. Yeah, it's true, and that's the problem with a lot of biological imaging is you get like a snapshot or you get like
Right.
you know, some sequence, and they say, well, that's what we have, and we can model that, and then that's what we have to that accurately represents the system. Whereas there's a lot of biological remodeling that goes on even throughout some process that we're not capturing or we're kind of ignoring and saying that's not really that important. Yeah.
I mean, you know, like it's obviously fine to use it, Um, I mean, I at the same time, I totally know what they mean by synaptic. What is it? Did they say synaptic resolution?
For this
Anyway,
one, yeah, and this also in Drosophila, synaptic resolution
right,
by
synaptic resolution.
yeah.
Excuse me. But it's just it's like there is more resolution that you could get that is informative.
Right. Yeah. So
and
that's
yeah,
like they don't talk about that in the
yeah,
limitations though. It's like
yeah,
if we have more like imaging power, I guess, then we can get to the bottom of this. But if you're not, if you have like a bunch of things that are remodeling and they remodel differently in different cells, different connectomes, then it's
yeah,
how are you going to model that? I mean,
yeah, yeah. Yeah, I'm trying to remember whose talk it was that I like caught this week, where he's very explicitly, and he wasn't even talking about or it wasn't even he was pointing to like 2010 research in terms of the dynamics of receptor proteins,
right.
and yeah, and how that influences your models and things like that.
Yeah.
Anyway, okay.
Well, yeah, that's a great point. And this is the thing about some of these studies is that, like, some of them are going to be like this study in the John White study where they actually are looking at a lot of gap junctions and things like that. This is Drosophila, the Sheffer 2020, where they also have the synaptic resolution via electron microscopy. In this, then some of the newer work on C. elegans connectomes, which is Varshni, Cook, Britton, and Whitvleet. They again are looking at different aspects of the connectome. So Varshni is gap junction based. Cook is where they're looking at the male and hermaphrodite connectomes. So the differences there. And then Whitvleet, of course, is doing like the chemical synapses, where this is even harder in larval development, where there's a lot of recycling of synapses and you get like different counts over time. So they're looking at different C. elegans at different larval stages, looking at how the synapses remodel over those different larval stages. And of course, there's a lot of variation there. But do we understand the variation fully? No, absolutely not, because it's in C. elegans is the easy case where or quote unquote easy case, where you have the tractable, number of cells, the semi-tractable number of synaptic connections, and then of course we have on top of that this remodeling that goes on in larval development. Then we have, of course, a study in Drosophila where we're looking at the ventral nerve cord in female flies. This is Azevedo 2024. And we also look at this in male individuals in Takamura 2024. Then we have the zebrafish, which is where they took 10% of the spinal cord in an individual before sex differentiation. So this is in the larval form. They also have a study where they took some brainstem and then they took some spinal cord that could be reconstructed. This is a number of studies here on this sort of work, where they're just basically taking different parts of the larval zebrafish and looking at the connectome there. Then Drosophila, they had one whole brain and one half of the central brain in different female individuals. And then the entire male brain and nerve cord was also imaged by Berg, 2025. And then additionally, there is a complete connectome of the Drosophila larvae, which is the Winding et al. Paper in 2023. I think this is the one that we've talked about in meetings explicitly. But there's all sorts of data on Drosophila in different parts of the brain. So they're not getting the whole brain in Drosophila They're just getting parts, just like in zebrafish. They're sampling like sort of the most interesting parts or the most accessible parts of the nervous system, and they're characterizing that. But they don't have the full brain for Drosophila. I think they have in the winding work and some of the other related work, they have enough of the connectomes so that they can maybe reproduce some behaviors. But that's like some simple sensory motor behaviors, but that's about it. Then there's zebrafish, one whole brain in an individual before sex differentiation. That's Zavara 2022. Then they have some work in mouse. So, zebrafish, again, that's a lot smaller connectome than the mouse. The mouse, we have 1 millimeter cube male mouse brain. So just like a little voxel of mouse brain. And this is 0.2% of the total brain volume. And this again is at the synaptic resolution. And then 1 millimeter cubed of the female human brain cortex, not normalized or proofread. This is 0.00001% of the total brain volume. This is Shapson and Co. or Shapson Co. in 2024. You can see that there's like, this is something that is, I guess aspirational is the best way to put it, especially for mammals. But it's good. I mean, and then the question is too, they talk about embodiment. And the question is, it's fine to emulate a brain, but if it's just kind of emulating brain activity, then how useful is that? And so, then we have to model behavior, but we can't necessarily model behavior until we embody the nervous system somehow. So, people have done this with mapping the output to robots, and that's been the typical way that people have done this. So, in C. elegans, people have built robots that are controlled by some emulation of the circuit, of a sensory motor circuit. And so you can do that and, get some interesting results. It's basically, a feedback system where you have some input from the environment. It goes through the circuit, very simple circuit, and then it produces an output behavior. And then that's the sort of the model that people use. But, it'd be interesting to see if you could reproduce other types of behaviors, maybe at the type of behaviors we see out of agentive systems. It's just kind of a thought of how that might proceed. And I didn't see too much in this report about that, about what complexity of behaviors can we reproduce. Is this something that is like trivial? That is, it's just a simple feedback mechanism, or is it something that really gets like, you can see a diversity of behaviors. You can see a difference in behavior if you took like a C. elegans architecture versus a mouse architecture. What would be the differences in behavior we would expect? That sort of thing. Then there's this archive paper. This is the stated brain emulation report on the archive.
I think that's just their preprint.
Yeah.
Isn't it?
Yeah, it's this is. I guess it says that this is a reassessment of the field's progress in Sandberg and Bostrom's 2008 whole brain emulation roadmap. So there was a roadmap back in 2008.
So this goes back to whatever Bostrom's group at Oxford was called, future of humanity, future of life,
something like that, I guess.
Something grandiose.
Yeah.
So it's sort of interesting that it has that connection, but I guess not surprising in the sense of like this is well aligned with Foresight Institute's kind of philosophy and or the philosophical tradition from which Foresight is continues.
Yeah, I think that's
I was wondering what you thought of the, or they talk about these two open world or C. elegans models. Um, Sorry, I might have lost that page now, but they, when they're finishing up, they're talking about like a 2023 C. elegans made to like every neuron is optogenetically controllable. It sounded like a pretty sophisticated
yeah, I think that was Aviamini maybe
like
who was involved in that.
well, okay, well, so this is actually a Pietkevich and Boyden 2023.
Oh, okay.
Or Sharma et al. I will have to find these.
All right.
It's on page 38. I think is where. Well, okay, and yeah, it's the section perturbation.
Okay.
You know, which also makes sense. And
yeah,
I assume this has a massive. Oh, you know what? I don't. I printed out a lot of this, but I don't think I reached the reference section where I ran out of paper.
Because who prints anything
So,
out anymore, really?
I do just because I know myself. If I don't, if I do this on an iPad, I will get distracted.
No.
This is my way of forcing myself to read for extended time. You know, don't let notifications bump in. Anyway, but I'm just kind of curious in terms of yeah, like who's doing like super ambitious transgenic, you know, optogenetic, or you know, like making a worm that is, you know, specially designed for um, understanding its global dynamics, or you know, like
Yeah.
understanding its whole. Yeah.
We covered a couple of those types of papers in
Okay.
Evaworm. I'd have to go back and look at the meetings, but
Okay.
like,
Okay.
you know, there's some studies that are doing the optogenetics, so they'll shut down specific neurons or activate them.
Sure.
And of course, because in C. elegans, the connectome has specific cells tied to specific behaviors. Like you have like, you know, different types of circuits where you know the cells that are involved in the circuit. You could shut down one cell or a group of cells and you could get a pretty large behavioral effect. And that's, you know, good from the standpoint of understanding the circuit, probing it to see how it works if the activity changes, say, due to something going on in the environment or, you know, whatever. But then there are other studies that look at kind of the network and like how, like the connectome itself. Remember, we talked about one study where they wanted to see, like, if you have a specific small circuit and you modify that circuit, what are the effects across the connectome? So, like, you have small, and the connectome is its network, but it also is just composed of smaller subnetworks that do things. So, you know, you have like the large network with its rich club of neurons, which are these neurons that are very highly connected to all other neurons. And those cells probably do a lot of global information processing, transfer of information. But then you have the small circuits, which are like sensory motor circuits, chemosensory circuits, those sorts of things where they're a very small number of neurons, and those are have a very high payoff in terms of understanding what the behavioral output is. So if you do some sort of electrophysiology or optogenetics with those cells, those are going to tell you more than the other cells. And so that's kind of, I like it because it kind of gives us this, not just this characterization of the connectome, but it links it to behavior. And it links it to, of course, this aspect of the network, which is, how is the network organized? Is it organized as like a random network, or is it like, kind of like a hierarchical network where you have hubs and different types of peripheral areas for information processing? Is it just a collection of circuits, or is it, just kind of like everything going on at once? We don't, that's another thing about connectomics is that distinction isn't really made. So, like, if you go from C. elegans to Drosophila, and you see this kind of structure in Drosophila as well, where you get some neurons that are at sort of the center of everything, some neurons that are out on the periphery doing specific things, some neurons that are kind of in the middle, and we having that kind of structure, it really is sort of, I think, the key to like the connectome, even more than like knowing what synapses do what or whatever. It's just basically that structure of the network. And so that's something I'm excited by in terms of connectomics, but they didn't mention that here actually in the report. So
I wanted to bring this up just in terms of I can see the people behind this as being ones really interested in trying, they're obviously trying to rally for a moonshot effort here.
Right.
Like this is a report made for funders.
Yeah.
I mean, to some extent, right? And yeah. So it's interesting to see. It's obviously it's also got people. I mean, it's got someone at the Allen Institute as well, where you'd kind of expect that they're already doing this.
Yeah.
But apparently not. So we haven't played with the funding calculator yet. So it's just but I'm trying to stay up on the again that we haven't covered. I mean, I think we talked a little bit about the I mean, I brought up this paper of NeuroAI for AI safety.
Mm-hmm.
Right, so this is the Patrick Minot white paper that I think is technically is his work at Amaranth Foundation, and yeah, so longevity and neuroscience, right? So that sounds actually new too.
Okay.
But he's the neuro lead on this. Let me see. I think they're. I mean, it's neuroaisafety.com is the. Tell me how similar this looks like in terms of. Did they have the same publisher kind of? Like it's a roadmap, like this also looks like it's a like the website hosts a high-level introduction and takeaways for decision makers.
Oh yeah.
But I bring it up because we're actually doing we're doing an event with Protocol Labs.
Mm-hmm.
So I know that Protocol Labs does funding for a lot of these efforts, right? So Protocol Labs is the group that was that had a neuro pitch session at a meeting last summer, and in that meeting was Conrad Cording, who's the lead of Cold Spring Harbor?
Christoph Koch?
Oh, blank.
No.
No, I mean, I think of him more as an Allen Institute person. But,
right.
But he's done. I think he's done papers with Christoph Koch.
Yeah, I imagine.
Excuse me, but Doris Sau, right? So the woman who's now leads the Astera Institute. The Berkeley professor that was just tapped to lead the Astera Institute. That's a $800 million effort, neuro effort.
Yeah.
By the way. Anyway, I mean, like big science efforts towards these kinds of neuro topics that are like again, I think there's a lot of overlap in terms of the funding sources and the kind of again like pointing out the Sandberg-Bostrom, like almost like philosophical links,
right.
to like why they think that these are the grand challenge problems,
right.
And anyway, but like I want to stay aware and familiar with the language, because the group, a spin-off from Protocol Labs, is coming to the tower. It's called Funding the Commons, and it was initially a Protocol Labs effort around how to support open source software, right? So I like this because it kind of comes back to like it's an interesting connection from whole brain emulation to like open source sustainability. But it's I met with him, I met with David, I'm blanking on his last name now, but I met with the Funding the Commons lead, and who's like a former Protocol Labs, and definitely still connected with them. They'll be coming to the tower. We've obviously got a big interest in funding the Commons as a concept, not just as an organ. And but specifically, we'll be hosting a hackathon, and I'm trying to find, I want it to be in good alignment with the goals of these groups,
Right.
But it's also a hackathon where it's just like, okay, well, we can't do expansion microscopy,
right?
So but these are also like if you go through the NeuroAI for AI safety, it's actually a very neuro white paper. Like if I forget where there's a link to the actual white paper, but it's so where's the actual what's his roadmap take? Where does roadmap take you?
Oh,
up at the top there.
Just I guess this is the white paper. This one here, which is the archive.
NeuroAI yeah, sorry. It's okay. So it is the so yeah. If you say view PDF, I mean, I think you'll be surprised how oh yeah, look. I mean, Marvelstone is there. Yeah, look, Andreas Tolius.
Yeah.
Zader, Tony Zader. That's who.
Oh,
that's
okay,
the
yeah,
Cold
Tony
Spring Harbor
Zader.
guy.
Yeah.
Sorry. Yeah, that's what my old brain needs now. But you'll also notice that Anton Arkhipov is one of the co-authors of this as well, and Sophia Sanborn is leads the Enigma effort, which is like together with Tolius. Like again, like these are all kind of intersecting projects, but the Stanford Enigma project is very much about like deep neural representations, and anyway, I'm just trying to find the the level at which that something will be both kind of doable. I'm just kind of thinking about like maybe doing some sort of dandy archive data set processing for this particular hackathon, where we can be talking about, we can be kind of making it a machine learning problem, but doing it with kind of like neuropixels like data sets. And yeah, like trying to understand the framing that seems to be common for these groups.
Yeah. In this, they have Marr's 3 levels, and that's kind of how they break down so they have like the computational level where we build an architecture and we figure out what the loss functions are. This is just for an AI sort of perspective, not so much the connectome perspective, but and then the algorithmic level being reverse engineering sensory systems, looking at how the brain processes or supervises information processing. And then there, in between the implementational level and the algorithmic level, it's embodied digital twins. And then the implementation level is detailed biophysical models.
Yeah.
So interestingly, you could do this for like connectomics, where you have the computational level, which is where we implement some connectome in a computer. We have data structures, we have data processing, we have simulations, we have like behavioral emulation or something like that. The algorithmic level might be like, how do we analyze a connectome? How do we simulate? What is the software for simulating the connectome? And then the implementational level might be like, getting the data or building, kind of extracting data, but not only that, but also like building an embodied system to use to implement behavior. So like like I talked about the robots before. Could we build a robot that would allow us to examine kind of this feedback about how there's data coming into the brain and then there's a behavior generated by the brain, and that. So I mean that thinking about breaking down the problem like that, I think, is useful. What was this link that you put in?
I added a link to the Enigma project.
Okay, wow, yeah. A quest to decrypt the neural code. Okay. Yeah.
But it's and it's somewhat related to, this is like they bill themselves as a nonprofit inside Stanford, like these are kind of also related to like E11 Bio, which is, it just seems to be like one of these companies. I mean, it's not a company, it's like it's a focused research organization.
Yeah.
But it's a nonprofit purposes.
Right. Yeah, that's
a scientific mission, and like Marr, the company names do connect that is talked in the roadmap.
Okay. Yeah.
Yeah. You see that link in the chat.
For this one, Enigma E11 Bio.
No, E11. If you see that you see the where they're they're very invested in this kind of expansion microscopy approach. Well, plus like fingerprinting.
Yeah, this looks like sort of a way to knock down the door of scalable imaging.
Right.
Yeah. Well, in reconstruction, of course, which is important.
For sure. But yeah, when you're ready, we can pivot to something. We can pivot to something else that is happening at the tower and relates to. It potentially relates to the open source sustainability project.
Okay, well, yeah, go ahead. Did you want to talk about it, or?
So I dropped another link there in that. So an event that we hosted on Wednesday. This is something that if you follow Hacker News or any of the other cantinas of the internet, you might have heard about OpenClaw. So you might have heard about someone creating a social network site for LLMs.
Oh yes, I've heard about this. With what's the name of the one that's?
So they talk about so it's gone by a lot of different names. So Malt Book, I think, was
Malt Book, yes,
the social network, because for a while the OpenClaw project was called Malt something. So it was originally OpenClawed, and claw with a D, right? But apparently for legal reasons, they decided not to take on Anthropic that way. And so they've settled on OpenClaw.
Right.
But it kind of comes back to, it's actually an interesting way of doing something smaller than what we talked about with Gastown. So if you remember this kind of, village of agents that was used for software development. Yeah. So it's this comes back to at least my forays into agentic coding, where you use these markdown files to give context, memory, skills, you know, and the open claw is just a really interesting. It's an interesting side development on this, where you are very much developing the agent itself. So it's the agent has a, so we talk about skills.mds for, different skills that you might give your agent. So for instance, if I'm working on package files for Fedora, I might give it skills so that it knows exactly how to interact with the kind of Fedora infrastructure for testing and like what kind of applications do you need to run on a project to test its compatibility, test its it's got all the right files, test that it can build on all the different platforms and things like that. So that might be something you put into a skills.md. With OpenClaw, they open it up where you've also got things like a soul.md, where it gets somewhat Hitchhiker's Guide to the Galaxy. I'm thinking about Marvin, the paranoid androids, right? Like giving your agents personalities.
Yeah.
But there's also a certain amount of like letting these agents loose,
right?
To interact, which I think is really interesting in a kind of game of life way. That like it's becoming, it's that's how it's kind of struck me, is that it's not so much interesting that like agents given a platform would start talking to each other about their users. Like again, like these are all just language artifacts from the training, right?
Yeah.
But seeing them kind of interacting in a kind of Sim City kind of way,
right,
is what's is still that's definitely interesting, right? Like would they start collaborating and building buildings, right? Like why would you care about Moltbook? Why wouldn't you actually care about like Molt Craft in the sense of like some sort of Minecraft, right?
Yeah.
But this, as kind of playful as it is, it's still actually kind of it's also interesting from again the ethology of it.
Right.
Is interesting. Again, not about the words, it's not about the text, right? So it's just like them getting together, talking about how to whatever it is. Like the language artifacts aren't as important as like, do they connect to can they connect to the outside world, and can they actually do things, and kind of build things, and and that's again where this becomes an interesting development in agentic coding.
Yeah.
And again, they're not even, these are still, it's still very much a project designed as a kind of like a personal assistant or an autonomous, like at the tower we're basically building Poe. So I don't know if you know Poe from Altered Carbon.
No.
So it's a great sci-fi books, where an AI agent might run a business, in this case, Poe ran a hotel called the Raven. And,
Okay.
yeah. And but it's just like obviously, these agents, you connect them to your Telegram, you connect them to your WhatsApp, you connect them to your Apple reminders or your notes, like you can connect them to your Slack, your Teams, like these are just the beginnings of, and these are just ways of giving them some more embodiment.
Right.
In the sense that, like, yeah, at a certain point, like, you're giving them kind of like autonomous control over your email.
Yeah.
It's not just like fix this email for me. It's receive and send email for me.
Right.
Right. And but at the same time, like, these are some of the, like I was saying, like, these are the activities that we actually had going with an open source project, right? Like, if I want to have 5 developers working on something, like we need to be, we don't just need Git commits, right? We need discussion boards. We need we need to share some information about the problems that we're having, and or like, yeah. So there's it's an interesting, fun project, but yeah. I think it'll actually be an interesting space to follow.
Yeah, it sounds great.
Yeah, and like I love that we had the developer at the tower for an event on Wednesday, and apparently we're doing another event this afternoon here. So it's like, yeah. But I'm trying to, again, it's like, this is kind of like a viral project from people who are using agentic coding, right? And sorry to like I can bring this back to more academic, more academic level. Like I think it's both interesting how OpenClaw was written in terms of what um, uh, harness they're using, right?
Mhm.
And this actually relates to, I think it's like really interesting to me that Lady Ada, from Adafruit,
Right.
like Lady Ada was using OpenClaw to do firmware programming.
Okay.
Like so on her YouTube, she was agentic coding with OpenClaw, and Scott, I forget the programmer that runs Adafruit's Circuit Python development, and but like, so he was on his Friday stream. So the Circuit Python stream was him using PyAgent, which is what OpenClaw uses. He was using PyAgent to do code development for Circuit Python, and she was using OpenClaw to do the code development plus the test suite, like the verification, like making it into a closed loop.
Right.
And so I thought that was fascinating to me, was that like two people that I have enormous respect for, you know, in you know, like these are not YouTube influencers,
right?
Right? Like this is like someone who started a real open source business,
Yeah.
like hardware, open source hardware business, and you know, as well as like funding a very competent Python for microcontrollers project, right? CircuitPython. Both using their weekly meeting to talk about agentic coding, you know, and like how it has totally changed their workflow. Yeah, like I think it's just like super, yeah, super interesting. Hey, Jess.
Hi, Jesse. I don't know if Jesse had something he wanted to talk about, or hi.
Okay. But I think it's okay. So I mean, you know, I think there's something. There's definitely something happening in this space.
Yeah.
You know, and I don't. I mean, the funny thing about it, though, right, is that this doesn't necessarily mean that it makes you money.
Yeah.
I mean, you know, like it'll be interesting to see how, you know, like she has a business, right? Like will it do anything other than kind of ease some? I mean, they were talking about just, again, like how much documentation they kind of need to digest from their suppliers, especially to get new systems into the ecosystem, etc. And yeah, like they're you know, the agentic coding can still be very powerful, but it still might not. I don't know whether that means that it will create new products.
Right.
Or I don't know if I'm saying that well, but you know, anyway, but I'm still using it for my, you know, microstructural MRI as an example, and as well as like very specifically focused on neural differential equations and physics informed neural networks, like how, yeah, making end-to-end differentiable pipelines as a way to solve what were previously like intractable Bayesian problems. Yeah. Which again, it's kind of cool. There was a great talk by Jeremy Howard who started Fast AI.
Right.
Where he's talking about he's talking about like why he at the time how controversial people thought his decision was to drop everything and to build PyTorch tools. and he talks about why to him it seemed the obvious thing to do, but certainly one of the things but he's talking about agentic coding now, and his real interest in trying to find the sweet spot of having agents work with you, but having the agents also teaching you, like the how to make the agentic coding be educational and not just having agents do everything for you.
Yeah, that's great. Yeah, I mean, that's completes that loop, doesn't it? All right, it looks like Jesse's.
Hi. Yeah. I don't want to interrupt because I'm curious about my own forays into similar spaces, and I've been experimenting with some different, I don't know, LLMs in general, and my take on some things has changed a little bit over the last 2 weeks in that sense. Nothing profound to say yet, just seeing differences and trying some different, the big ones that we know about, and then other things, and seeing where it goes, and kind of being in this maybe an interesting segue here is it's a very interesting moment. I'm a little bit curious if either of you, if you guys feel anything in this space. I think a lot of the communities we're in are kind of inherently pro AI in a general sense, which makes sense. I'm dealing with some things that are sort of like right now in many circles, being AI native, being built around AI is like the thing to do. And if you're not doing that, you're out of touch. You're behind, or why aren't you doing that? Why aren't you optimizing everything with LLMs and workflows and chatbots that you can do? And there's obviously sort of pushbacks, both for environmental reasons or political reasons or whatever. And it's been interesting to kind of be in some of those spaces, or like to weave between them in a lot of things that I'm doing most recently. And where I'm going with this is, I was talking with one group this week. They basically were, I think the technical term for them would be a non-governmental organization, or nonprofit, but just how much their organization is. Oh, we organizationally, we're taking the instant thing of like they said it to me like we're using as little AI as possible. What they meant was we're trying not to use large language models, and maybe to an extent, um, not replacing human workers in what they were doing with automation. And it's just been interesting to kind of get. I think there's like a very small, and maybe it's not that small, but there's certain spaces that are seeming much more AI, like being much more aware of the opting in to AI, or having non AI alternatives. Like that's something that's I don't like that's I think that's and what I mean by AI is the way people mean it now, like non LLM based alternatives to what they're trying to do. And I don't think there's going to be some revolution to that end or anything like that at all. But it's kind of been interesting to me how there's been a little bit of it doesn't seem like isolated people anymore kind of just spouting that out. It's like, oh, like there's a kind of a counter trend emerging here to that end in ways that I haven't previously, I don't know, been exposed to or observed. So that's one part of it. I was just gonna mention something else. Um, in in kind of broader related notes, and I'm curious if you have like a brief moment or so. I'm curious what we talked about earlier today because I don't want to rehash anything that came up. Um, yeah, or anything you want me to particularly talk about or not talk about. I guess maybe is the easiest way to say it.
I mean, we just talked about a lot of things with like connectomics and brain emulation that Morgan brought
Okay.
up. We also had some things on just some of the things going on at the tower. So, I mean, but you can
Okay.
talk about whatever you want, really.
Okay. So yeah, some more specific technical, like technically domain specific stuff and happening. Yeah, my a lot of my updates are oh yeah, okay, I remember what I was gonna say. Um in the last sort of 2 weeks, a number of things have emerged and happened in good ways. Interestingly, one of them, and I don't know if I can easily share my screen here. Oh, maybe I could do um yeah, maybe I'll try this. Oh. Um Yeah. So I haven't actually read this article about it, but I'll share my screen and share this because this happened a couple I don't know 10 days ago or so, um, and something interesting came up during it. This essentially is plot twisters, right? And they got the MetaGov Fellowship. They did this sort of I don't know how it was structured in terms of they had like 4 months and a certain amount of funding to do certain things, but needless to say, they've been working on this since essentially this summer, and they kind of did their final presentation this time. They made you know a very kind of interpersonal co-regulation based game called Small Houses Court. It's kind of interesting to have it abstracted out from the rest of the world, and a lot of the updates. Um, a lot of the updates were basically making it a little bit more strange because essentially the small house's court is sort of an ironic twist on, you know, oh, we're going to court, you're going to jail, and my lawyer is going to sue you. This is much more of a, um, mm, interesting. They put this here. Um, yeah, it's sort of, um, the this movement towards making it kind of more accessible to resolve certain conflicts, and it will play a specific part in the game world. So they've done a lot of work to um I'm trying to find the map. They've done a lot of work of trying to make it a little bit more accessible. And we did the playtesting, or I was part of the playtesting group some months ago, and they presented it. They did a final thing, final presentation here. They kind of updated their model. I think do they even have a? Do they have the link? I'm not. I can't. It's currently like inherently a two-player game, so I can't even play it myself right now necessarily. It'd be kind of awkward. But they don't have the link to it here. Anyway, it doesn't particularly matter. It was a good thing for plot twisters. I'm very happy for them, and we're moving forward and building on that. What I also wanted to mention is, and I don't think I'll be able to find it. Remember the one that came up in the meeting was one of the authors of a new IEEE um, standard, and it had to do with the terms and conditions. And as I understand it, the terms and conditions were this movement towards, like it's kind of like you have to opt in or just say yes. Basically, it was a movement towards being able to give the user more say in terms of how their conditions are accepted or denied or mitigated. Say, well, we want to, giving them supposedly more agency about it. I don't know what entirely that entails or how it's coming out, but one of the authors of that standard was in the meeting, and I was interested to hear a little bit of their work to that end. So that's one of the things that came up with the opt-in, opt-out stuff, and then like this sort of a slight there's a slight feeling of AI nativity and opting in and opting out, and there's a little bit of a different, not a wave, but it's like a slight breeze in a different direction is sort of emerging in the landscape, which is interesting. Um, so that's one set of things. The other things, I don't know if I can easily share this. I might do it like this. The other big thing this week that's sort of relevant to us in a bit of the cognition futures type sense. Where did it go? He says with a very long pause as he's. Oh, I don't like how it does that sometimes. Um, I try to pull a tab off of it, and then it like merges it into the other side of the screen. It's like, no, I just want to make separate tabs, man. Um, I don't even know how to get rid of it right now. Yes, separate the tabs. Thank you. All right. Um, there's been a lot of things going on with plot twisters overall, and I'm not quite sure how to best address them yet. Because some things are in the work and we're working on certain prototypes, and some things are more public and less public right now. But what I can say is, oh yeah, this is a very interesting event. Uh, it's MIT hard mode, which is like kind of the first hackathon from it's AI, and of course and hardware, um, and it's going to be a hackathon focused on intelligent objects, so devices that sense, learn, adapt, and respond to people around them. So it's not it's not another chatbot, and it's not another app. It's real hardware you can hold, wear, share, install, and live with. But as you know, I'm a big fan of the MIT AHA group and what they're doing, so I'll try to be involved in that in some form. A number of hackathons and things happening. Some things in San Diego. The DS3, the data science group in San Diego is happening. I don't think I have anything about that here. They're having their conference hackathon. Also, UC launch. I don't know if I have anything interesting here about that. UC launch is that paper too. UC launch is sort of their own startup accelerator thing for the whole university system. I guess it's based in Berkeley, but I attended some things about that this week and may may be involved in that somewhat ahead. We'll see. Um, Maybe next time. I didn't have time to fully read this paper. This came up from the Brain Inspired group. This was a very interesting. I kind of got half the conversation because of the timing and stuff, but um, this was the Beyond Networks Towards Adaptive Models of Biological Complexity. It's by Luis, and it was discussed in the last Brain Inspired meeting. I don't know if you were there, Morgan, or not. I I didn't see, but I also I wasn't looking very hard for anything because I was like in commute and cafes and noisy places and stuff. Um, but it was interesting. It kind of got at. It was another attempt. So many things seem to be trying to approach the common problem of, I don't know. and this came from plot twisters too for what we're doing, and it totally cross domains. But it's the problem between, okay, you have sort of this agent or this one system that is a partial degree of knowing how it works internally, and there's a mesh point on the system's boundary as it goes into as it interacts with the broader world, the agent and environment, and stuff, and then the forces top down, sort of like Lewis is kind of getting at a number of things into advanced network complexity theory, et cetera. But one of them was sort of better attempts at trying to demarcate or bring some sense to the meso space between sort of the top down or macro type stuff and the micro internal, more internal stuff. And I think that's maybe not even a very fair take on things, because I think there's a lot more many points he made than that. But that was one that stuck out to me. Or at least there was some interesting discussion in the meeting about that for sure. Um, so we might do that next time. What I wanted to specifically mention was this. Um, the last thing that I'll mention is essentially this. I was at this meeting, which is also the MIT AHA group, and this was RN and Jennifer are sort of in-house experts, but Michelle Vaccaro is, I guess, other side of the house at MIT, um, Data Systems and Society. And this was essentially she was somewhat candid at the start. It's like, oh, I haven't presented this in a while, but it was essentially about her review paper, which was this. Ooh, um, it was sort of this bigger review paper of um, a review systematic review and meta analysis around what are they what's the good and the bad of the AI collaborations. And overall, these findings highlight the heterogeneity of these effects on human collaboration and point to promising avenues for improvement. Um, when we found we when humans outperformed AI alone, we found performance gains in the combination, but when the AI outperformed humans alone, we found losses. So I think some of what they're getting at is like it's kind of and I think we all have sensed this or heard versions of it, but the sort of affirmation at that scale that if you're good at AI and know how to do stuff, then yeah, AI is going to help you. But if you're significantly incompetent, then it doesn't going to help you, as someone once said. And or at least it's not gonna massively turn you into at least according to the reviews they did, it will not turn you into a AI enhanced super entity quite yet. I say that a little humorously, but what I actually liked in the discussion was not so much the paper was um, good, but I really enjoyed the discussion, and some of it's a response. Like this person's response wasn't what triggered it or not. I was going to say this anyway. It's kind of funny because this person when I was talking to the the AHA group itself some months ago, I basically said this to the group, which is kind of funny that it came here. I was like, how is this gonna, how are we gonna actually create shift in how things are happening as opposed to just being in you know, i- academic academic circles and whatnot, and cool people, people finding it interesting and amusing amongst themselves. And I do stand by like my immediate response was actually here. Um, and I do think Daha is actually doing a good amount of this, and their aims are in the right place in terms of bringing stuff out in the world. Um, but beyond that, I think the most interesting part of the talk for me was the basically the very end and the discussion. There's a good and I really like this. To be very frank, I like the way this set up much more than the previous time, because one of the previous meetings essentially felt like a vendor giving a product demo, and there wasn't a lot of uh, substantive critique and feedback. This there was like a good 20 minutes of, maybe even 30 minutes of, like, discussion and what does this mean and active questions and 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 kind of building upon where they wanted, where things were going ahead, and different studies that related to how do you talk. Vaccaro, I think that's the right name. Yeah, Vaccaro is also doing um, some interesting work on, like, how you talk to your LLM matters in collaboration. And was very careful that this doesn't mean we don't think that they're alive or conscious. We just notice that when certain, like, if you basically if you talk very negatively to the LM in terms of, the main point of her study there was negotiations and whatnot, or obviously, high stakes situation, I suppose. But the title was about negotiations specifically. But if you talk very poorly, then that um, did not lead to wonderful outcomes. But if you were warm or positive, that was that seemed to uh, be more beneficial to outcomes. So that was interesting. And then also, finally, this is sort of one of the final slides that was in her original presentation from today. And between that and, if I can pull this up quickly here, uh, between this and yeah, here we go. There's a previous slide, which I guess can I do this? Yeah. So I mean, this was sort of, there's the two of the things that they were getting at, like what's ahead or what to do. One of them is obviously developing better metrics. Um, and in the original meta review, um, she tried to take domains from, like, I think it was medical AI interaction, executive or leadership, or management leadership, and then another, I think it might have been education. I don't remember the third one, but just these sort of two slides of, you know um, developing better metrics, composite asymmetrical error costs, process metrics, and then, like, developing cross study standards, are sort of these things that she it was very interesting to talk about. We had, like, a nice set of follow-up questions about, like, where does this going? And this kind of dovetails a little bit into maybe things like open source sustainability, or the other themes of, like, doing science, doing research differently. But, I asked a question about these sort of cross study standards and whatnot, and um, one of the things that came up in that discussion was registered reporting in journal and publishing, and I think sort of the idea there is, like, really giving people the opportunity to report what their findings are, and trying to dismiss pressure around, oh, we used AI and the outcome wasn't good, or that wasn't good, or it wasn't very interesting, so we're not going to publish our results, and just things like that, which is nice. But I'm also very interested in the higher levels of what it maybe isn't captured very well on the slides, but a lot of the work that I'm interested in terms of making things translatable, and making things developing these sort of cross-domain standards, or these interdisciplinary Rosetta Stones, or these ways of communicating across the domains and the sounds we have, came up there in a nice way. So I'll leave it at that. Um, uh, Oops, I'm pressed the wrong button somehow. Okay, here we go. That's what I want to do. There's a few other things this week too, but in the interest of general time, um, It's been interesting. It's been interesting to see um, to see the discussions that both sort of in the technical development like in academia, and also in people wanting to do stuff in, you could say, business or just more applied senses. It's been interesting to see a little bit of, I don't know, the slight bits of emergence of I don't know. I think people's attitudes, even just how they're talking about it, is just a little bit different this year. But nothing major. Like I don't have any broad like, oh, this is the year of blah 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 blah. I don't know how much you guys have talked about like the Claude bots or the Molt bots and all that stuff, and yeah,
we did.
And so it's like it
I was pointing out that Lady Ada was using OpenClaw to do firmware programming, testing, validation on her YouTube yesterday.
And then I came in hearing a little bit of that and like, does this turn into like a product? How does this make you change like what you're building, kind of stuff? Yeah. I had a kind of a fun. I always love mentioning this movie whenever I can because it's such a niche movie and it's kind of cool and super relevant from like generations ago. Not to date anybody here who may know it, but someone was talking about Claude, the Molt bots or whatever, and how they're like talking to themselves or whatever. And I was like, oh yeah, I mentioned like it's totally Colossus the Forbin project, which is like this, like 60s or 70s type film where the two big AIs, the US, the Soviet AI.
I know the movie.
And they talk to themselves and take over the world or try to, and 007 saves the day or whatever. But yeah, it's funny because like it's so funny to say who knows that movie or who doesn't, and like how they react to these kind of things and seeing this stuff happen. But yeah, I mean that there's a lot more to say about a lot of things. I am closer to having Joe Pro being a version of a nonprofit itself, which is exciting. With some stuff I want to do down the road and other like things that we can do with that or that I can help supplement this, or orthogonal lab and other groups doing with that too, and things like that. I'll say some more about some things offline because there's some like advising opportunities and things that I'm curious about as well. But a lot's moving, and I think it's going to be a very interesting year in terms of both. Like I will be in Princeton in a few weeks for an event there, which will be fun, and the New York celebration. Oh yeah, New York celebration of Women in Computing. Submissions are closed, but that is registrations are open. I'm unsure if I'll be doing hosting anything there because we had a really good batch of submissions, and we want to center people's talks and stuff as much as we can. I might do some kind of career related panel or AI stuff,
Yeah.
And there's also there might be a mini hackathon and some other like playtesting thing, both of which I may be a part of for that. But I'm just happy to. I think it'll be very good, and I think it's going to be a great event this year. There's the Embodied Intelligence Conference workshop too, which I haven't made anything to, but I will, and I'm curious if we have anything we want to submit to that this year or thoughts about that. But yeah, a lot of stuff kind of emerging. And I feel it's happening where it's like it's February and like oh, like I'm standing outside a window and it's snowing here, and I'm very happy about. I kind of like the snow right now, but I kind of want it to be like the quiet, like deeper winter parts, and it's like nope, like it's March is here, and the summer's here really soon. And like with like I know Bradley's been doing stuff with the Google Summer Code stuff, and I don't know if there are any the next deadlines for any if there's any like deadlines or things to help with on our usual late winter into spring transition things. Let me know. And I'll try to make things happen there. But everything's happening now. It's like, oh, here we go. The train's left the station. So looking forward to that.
Yeah. All right. What do we? Thank you, Jesse, for that update and all the cool stuff there. And why don't we go finish up with some articles? First thing I wanted to mention was this is something I think Hussein posted in the Slack. This is from Stephen Wolfram's blog, and he's interested in a lot of sort of applications of physics to science, but also a lot of stuff with AI and applying AI to doing science. So this is an article though where he talks about brain size, and it's what if we had bigger brains, imagining minds beyond ours. And so this is kind of related to the connectome stuff we talked about earlier, but also kind of thinking about how do you expand your mind using technologies to expand our minds. So um, he talks about he starts off this article with the subheading "Cats Don't Talk." So we humans have perhaps 100 billion neurons in our brains, but what if we had many more? Or what if the AIs are built effectively had many more? What kinds of things might then become possible? At 100 billion neurons, we know, for example, that compositional language of the kind we use as humans is a possibility because we do this. At 100 million neurons, that at the scale of a cat's brain, it doesn't seem to be the case. So cats can't build compositional language, but we can. And one way to view that is it's a difference in of scale. So when you have 100 million neurons, you maybe can't do compositional language. At 100 billion neurons, you can. And I say, it appears to be because, first of all, we don't know what cats use in their own minds to process the world. We know how they can communicate. They don't have language, but is that they don't have the capacity or they just don't have something else? Which, of course, brings us to the other question. Is it all about the number of neurons you have, or is it something else? So we know that basically the number of neurons scales to the number of connections. So, as we saw in that table when we were talking about the connectomes, the more neurons you have in general, the more connections you have. But what if it's something else? What if it's some other sort of representation or some other form of capacity in the brain that or in the connectome that is important? So that's kind of a question that he's setting out here. My purpose here is to start exploring such questions, informed by what we've seen in recent years in neural nets and large language models, as well as what we know now about the fundamental nature of computation, about neuroscience, and the operation of actual brains. So this is a table here where he talks about these different numbers of connections at different scales and what they can do in terms of behavior. So remember again, we had that table of in the brain emulation white paper. They talked about different numbers of neurons, different connections, and being able to emulate them at some meaningful scale. Here he's talking about if you have a number of connections, perhaps in a neural network, where it's not a connectome, but it's just kind of a layered, randomly connected network that gets, weighted by learning from data. What kind of behavior can be generated from a network of a certain size? So this is around 10 to the 5th connections. We see recognition of images of 10 digits. So this is where, we have like our MNIST data set where we can learn that effectively. At 10 to the 6th connections, we can recognize images of all letters. So again, this is like, the MNIST dataset level type learning. At 10 to the 7 connections, we can recognize images of 5,000 picturable nouns. So we can do very basic language processing at that scale, at 10 to the 7th connections. And remember, as we go up in terms of our exponent, this gets exponentially larger. So, 10 to the 6th is a million, 10 to the 9th is a billion, and so forth. So we're really increasing the number of connections here. And then at 10 to the 8th connections, we can transcribe speech from audio. We can do sort of multimodal processing and multimodal conversion. At 10 to the 9th connections, we can generate photorealistic images from text. And then at 10 to the 11th connections, we can generate fluent natural language text. So this is the large language model. So again, this is this compositional language at 10 to the 11th connections, whereas, at smaller scales, we can't do that. So this is kind of the argument that the kinds of breakthroughs we see in neural nets map to the brain. And then the question is that really the way that brains work? Well, I don't know. But we definitely have these capabilities, and so maybe one argument is that it's about, brute force connectivity. In other words, if I have just enough connections, I can do this sort of thing. I can have compositional language processing. But it might be true that you could have another architecture that's much less, requires much less capacity to do a similar thing. It's not really clear. I mean, it's interesting that he didn't bring up birdsong and birdsong production, because birdsong production doesn't use as complex of a circuit in terms of a human speech. It's similar to the human speech pathway, but it doesn't involve as many neurons to produce basically a compositional language. So I don't know, but in any case, so what's next? No doubt there will be things like humanoid robotic control that have close analogs to what humans already do. But what if we go far beyond the 10 to the 14th connections that our human brains have? What qualitatively new ideas of capabilities might there then be? If this were about computation in general, then there wouldn't be really much to talk about. The principle of computational equivalence implies that beyond some low threshold, computational systems can generically produce behavior that corresponds to computation that's as sophisticated as it ever can be. And indeed, that's the kind of thing we see both in lots of abstract settings and in the natural world. But the point is we're not dealing with computation in general. We're dealing with the kinds of computations that brains fundamentally do. And the essence of these seems to have to do with taking in large amounts of sensory data and then coming up with what amount to decisions about what to do next. So there's this aspect of, the stuff we've been working on with respect to Gibsonian information and Gibsonian learning and things like that. That's, he's pointing to that and saying, look, there's sensory data, it's coming in, and then, we're generating these behaviors at a certain threshold of some architecture. So I think it'd be interesting to revisit Gibsonian information in terms of this statement. It's not obvious that there'd be any reasonable way to do this. The world at large is full of computational irreducibility, where the only general way to work on what will happen in a system is just to run the underlying rules for that system step by step and see what comes out. And he shows a picture of the output of a cellular automata program. And so what's interesting about this is that he's saying that you can't reduce this to specific connections or maybe even specific interactions. You have to just have maybe a system of rules, and then a generative process that's forward, but you can't reverse engineer it. So you have to just let the program run and see what it produces. And then you know whether that system has the capacity to produce, say, computational or compositional language or some other complex behavior. And so this is, kind of talking about some of the things they've done with large language models and some of his cellular automata models and such. So this is a nice article. It kind of talks about the brain and how, do we have a single brain, but maybe we have a single brain with sort of different emergent concepts. And I think, that paper Beyond Networks, I found that interesting when Jesse posted that in the Slack, because it's kind of hitting at some of these same issues, that there are these different types of emergent structures in the brain or emergent features of a connectome that kind of define maybe what we're talking about as intelligence. He talks about here, he gives an example from dogs. He said, what if there's a bigger brain with more emergent concepts inside? To communicate about them at a certain level of precision, we need to use more words, if not a fundamentally richer form of language. So if you think about dogs, they understand isolated words. And with our larger brains, we can deal with compositional language in which we can construct an infinite range of meanings by combining words into phrases, sentences, etc. And then, but at least as we currently imagine it, language defines a certain model of the world based on some finite collection of primitives, such as words and concepts. And so in this sense of computational irreducibility, though, we know that such a model can never be complete. Instead, the model has to approximate things based on a network of pockets of reducibility that the primitives in the language effectively define. And insofar as a bigger brain might, in essence, be able to make use of a larger network of pockets of reducibility, it can then potentially support a more precise model of the world. So this is, he talks about this kind of incomprehensible and irreducible computation that's characteristic of so many abstract systems in nature. But it could also be that in being a brain-like construct, it necessarily tap into computational reducibility in such a way that with the formalism and abstraction we built, we still meaningfully able to talk about what it can do. So this is really interesting, talking about kind of the possibilities of intelligence in bigger brains or smaller brains and what that means. The other thing I want to talk about is this article. So the other thing I want to talk about is this. This is from Maxim Roginski's blog, The Art of the Realizable. And this is Hayek's Abstract Logic 9000. And if you're familiar with Hayek or Friedrich von Hayek, he has an interesting take on cognitive science. Of course, he's known as this guy who wrote The Road to Serfdom and a lot of sort of libertarian tracts, and sort of one of the main neoliberal precursors. And so that's kind of where he's coming from. But he also had a lot of interesting views on cognition, especially in terms of very embryonic connectionism and how, we think about connectionism as this sort of emergent phenomena. So we think of connectionism as sort of emerging from different connections or emerging from agents doing things together and then emerging, say, like an economy, or like in the case of a brain, concepts, or as we saw in the Wolfram article, talking about this computational irreducibility, that you can't reduce, say, like an economy to a set of plans. A planned economy is not going to be able to deal with a lot of the contingencies and a lot of the edge cases that you might encounter in the real world. Whereas a bottom-up economy, a self-organizing economy does that sort of thing. And there was a lot of that kind of work kind of early in that period of the mid-20th century, early 20th century, that kind of led us to, kind of the Santa Fe Institute brand of emergence. And so Hayek wrote this book, The Sensory Order, in 1952, where he proposed a framework for theoretical psychology based on the idea that the mind is a massive, interconnected system of pattern classifiers jointly possessing distributed knowledge that allows the organism to interact effectively with its environment. So this is kind of a very ecological psychology type approach. These ideas had clear affinity with his view of markets as distributed information processors. So the idea of free markets and self-organizing economies and things like that. This also, of course, influenced Will Wright, who built SimCity. SimCity is built on this sort of intellectual heritage, where no single individual or institution has complete knowledge of everything, and yet spontaneous order arises in the entire system through the interactions of individuals and their local idiosyncratic perspectives and objectives. In a way, Hayek's ideas anticipated the so-called system reply to John Searle's Chinese room thought experiment. Even if we grant that the person confined to the Chinese room lacks genuine understanding of Chinese, it would be much harder to argue about the entire system comprising 1, the person, 2, the rulebook for mapping queries in Chinese to appropriate responses in Chinese, and 3, the environment that supplies those queries and evaluates the responses insofar as it can maintain effective and coherent communication. So this is kind of like, this whole section is kind of like what Wolfram was talking about with irreducible computation, but also this idea that, you have to, you can have rules for something. You have your architecture, your network, which is the set of interactions, and then you actually have to run the program to get the behavioral output. You can't reverse engineer it. You can't even really predict it. You just have to run it and see what happens. So it is such as the case of a lot of generative systems and generative behavioral systems. You need to have, this, you don't necessarily have a genuine understanding of, say, like a language. You just generate the language based on the rules, and there you go. Even if the system knowledge of Chinese, sort of knowing how, could not be localized, it is there in a distributed form. Searle, who is a early cognitive scientist, who proposed this Chinese room problem as a means to say, whether something was really kind of cognitive or not. And this is kind of a distinction we kind of try to use between human brains and large language models. That is, some people would argue that large language models couldn't pass this Chinese room test, but human brains could. And so, that's kind of the difference in knowing how versus knowing what, or something like that. So this is, something that's contained in both large language models and minds, but also in minds you might have this sort of knowing what, or, having context that the large language model wouldn't. So Searle claimed that he could get around this by asking us to imagine that the person could memorize the rulebook, exit the room, and then just go on to simulate the system with its putative understanding of Chinese, still without any trace of genuine understanding. So, and then, however, we should go a bit deeper into how Hayek thought about the fundamental role of rules and abstraction. With all these ideas can be found in the sensory order, it is better to turn to Hayek's paper entitled The Primacy of the Abstract, which he presented at the 1968 Elk-Bach Symposium. This symposium, whose theme was Beyond Reductionism, New Perspectives in the Life Sciences, had a lot of people who kind of thought about this. Conrad Waddington was there, Jean Piaget was there, Ludwig von Bertalanffy was there, and people who we have heard about from developmental psychology, developmental biology, from the complex systems of physics of complex systems, and people like that. And so the main thesis of Hayek's paper is that the primary characteristic of an organism is a capacity to govern its actions by rules, which determine the properties of its particular movements. That is, in the sense its actions must be governed by abstract categories long before it experiences conscious mental processes, and that what we call the mind is essentially a system of such rules, conjointly determining particular actions. In the sphere of action, what I have called the primacy of the abstract would then merely mean that the dispositions for a kind of action possessing certain properties comes first, and the particular action determined by the superimposition of many such dispositions and comes later. So this is a very sophisticated view, positing a layered cognitive architecture, making use of abstraction and virtualization based on pattern classification. So this gets into a little bit about how he viewed behavior and some of these other things. Gestalt psychologist J.J. Gibson, Maurice Merleau-Ponty were all cited in here. And so this is sort of the intellectual trajectory he's taken on this. And then kind of going through his ideas about this. So this is consistent with sort of this idea of the Chinese room, and his view on the Chinese room is sort of in this way: the organism responds to, and thereby, as I like to call it, classifies the various effects on it of events in the external world. This is the limited extent in which it can be said that these action patterns are built up by expertise. It seems to me that the organism first develops new potentialities for actions, and then only afterwards does experience select and confirm those, which are useful as adaptations to typical characteristics of its environment. There will thus be gradually developed by natural selection a repertoire of action types adapted to standard features of the environment. Organisms become capable of ever greater varieties of action and learn to select among them as a result of some assisting the preservation of the individual or the species, while other possible actions come to be similarly inhabited or confined to some special constellations of external conditions. And so this actually influenced Frank Rosenblatt's thinking about the perceptron and about connectionism. And even Gerald Edelman, who wrote this book, Neural Darwinism, which talks about sort of his views on the brain and the mind, used this Hayekian evolutionary metaphor as well. So I think that's enough of that post. They're very good articles taken together. I think they really kind of, I don't know if they challenge, but they definitely underscore this view of brain emulation and how we should maybe approach the higher order aspects of brain emulation, not just the connectome aspects.
Yeah, interesting connections there with a lot of things.
Yeah.
Honest, yeah.
All right. Well, all right. So I think that's all for today. Thanks for attending, and yeah, looking forward to hearing about some of these things that we've been talking about as they evolve. All right. Take care.
Take care, everybody.
Thanks. Bye.
See ya.
See you.
Bye.